Today is chapter 17, and we're going to be finishing up our special senses. Yeah, so this will be the end of chapter 17. And we kind of did the eye anatomy. Well, we didn't kind of. We did do the eye anatomy on Tuesday because we kind of covered it in lab. I believe this is where we left off last time, where we just kind of talked about each of the things we talked about, and then we kind of reviewed it. So, vascular layer, we have our iris, which contains our blood vessels, our melanocytes. What do the melanocytes do? They do our melanin, so our color, yeah. And then we have some smooth muscles called your pupillary muscles. What do you think our pupillary muscles do? Yeah, they affect the size of our pupil. What is the purpose of changing the size of our pupil? Yeah, it's a light control. So if we open up our pupil, we're going to be letting a lot more light in. But what if we're outside and it's super, super bright? Do we want all that light flooding into the back of our eye? No, we're going to constrict our pupil down. So these muscles are basically controlling that diameter at the iris level of the pupil. And your pupil is that central opening of the iris. And we talked about in lab, the iris is the colored portion of our eye. So if I look at you and say, oh, you have blue eyes, you have brown eyes, you have green eyes, that's your iris I'm looking at. Okay, and this shows your pupil. You can see we have, you don't have to know these individual muscles. They're just all part of that pupillary muscles. We have one that dilates and then one that constricts our pupil. And it's all based on the amount of light coming into the back of the eye. All right, ciliary body. This is still part of that vascular layer. It attaches to the iris. We had our oris areata. What was our oris areata? Yeah, it's the lace in point where our retina, which is our neural layer, laces in kind of to that vascular, that iris ciliary body layer there. We have some suspensory ligaments called the ciliary zonule, and these actually attach out to the lens, to the ciliary process of the lens. And basically, this is how we change the shape of the lens. So I talked about refraction, all of our ability to see clearly and adjust long distance to short distance is based on how we refract the light coming in. What was the number one thing that refracted our light? The cornea, right? And then what was the second main thing that refracted light? The lens. So the lens does kind of the fine adjustment. The cornea is going to bend the light, and we're trying to bend all the light to hit near that fovea centralis, right? Because that's our main focal point. So our ciliary process, or the ciliary ligament out to the lens, can cause the lens, when it contracts, the lens gets a little rounder. When it relaxes, the lens will elongate. Anybody need more time? All right, choroid. The choroid, this was basically that dark layer in the back. We saw the tapetum lucidum on it in our cow eyes. Choroid's a double layered tissue. You have a fibrous outer layer, and then you have more of a vascular layer. You're going to see a lot of capillaries and blood supply go through the choroid, which is going to deliver oxygen and nutrients to the retina. It makes the very back portion of the eye, and our light is going to go in, and it's going to kind of going to come off the choroid up to the retina. Remember how I kind of last time at the very end said how the light flows all the way through and then comes back at the retina? So the retina actually receives the light back off of the choroid, not as it comes through the light to the choroid. All right, and our inner layer of the choroid is that retinal layer, and it absorbs our light. And we have our photoreceptors in there, which is our rods and cones. Is everybody good? All right, the rods do not discriminate colors. It is our cones. Remember I told you C for cone stands for colors. That's not really what it stands for, but that's how you can remember it. Rods are just really sensitive to light. We really collect a lot of light from our rods that are dispersed throughout. We're going to see our highest density of cones in that macula, as we talked about in lab, that macula luda, macula densa, you'll hear those terms for it. And that fovea centralis, which is our focal point. It is the center of the macula. And this is where we have our sharpest color vision, and we're going to see a lot of detail in those particular areas. So our visual axis is pointing at that fovea centralis, which our visual axis. We're bending all our light, trying to get all of it to shine right onto that focal point. If we're not refracting the light in a correct way, we can have our image fall short or go too long, and that's going to be our nearsighted, farsightedness. Was everybody good? Okay, so this shows kind of a cross section. This is going to be a back pigmented layer of the retina. We'd have our choroid back there. Here comes our light. And then it collects, it goes from the back up. And this is the first place where you receive light, but this would be the front of the eye up here. So it comes all the way through, goes to this pigmented layer, and then it gets received up here by the rods and the cones. The rods in this picture are these little brownish looking cells here. The cones are these purplish ones. So you can see we have a lot more rods than we do cones. And these are the sensitive to light. These guys are my color. And you're going to see, we're going to talk about some of these cells in here. Here's a horizontal cell, a Macerin cell, and then our ganglion cells. What's a ganglia? Cell body, right? So what we're going to see is we have a whole lot of these guys collecting light or information. If it's a cone, they're going to come in synapse. And by doing this, we're kind of beginning to pull all our information into a more centralized location. So these are slowly pulling this information together before it synapses and then goes to the brain. And this is just a microscopic view of that. This shows your vessels. This is, remember when I was talking about the Optomap you can get at your doctor's office? This is almost exactly what it looks like at your doctor's office. This would be your fovea centralis. And usually all this kind of tannish orangish in here is your retina that they're looking at. And then you can see that vascular layer of it with all the light coming in. Yeah, and this is the optic disc. So this would be our blind spot right here. We don't have any of those neural receptor cells right where the nerves come in. Why? Because it's also where all the blood vessels come in. So you can see all my blood vessels are coming in at that point. And this is an illustration of it. So right here is where we were looking at where all the blood vessels are coming in. And there's no receptors there because the vessels are across there. So that's why this is your optic disc or your blind spot. And then your neural tissue goes this way. And so light's going to go all the way through to the pigmented layer, and then it's going to reflect back and then come in like this.
It's not crazy, so it's probably immediate, but we see it as
yeah, we do, because it travels so fast. But yeah, that was one of the most interesting things. It just travels through, and then we receive it coming back towards the anterior portion of the eye. Okay, and basically on these neural layer cells, remember I was showing you they were the ones that begin to kind of pull that information together. Your bipolar cells synapse with rods and cones. Your ganglion cells synapse with your bipolar cells. Your horizontal cells, those are kind of the first ones we saw up under the rods that come across. And then these guys that look like horizontal cells as well. I'm not going to quiz you a bunch of information on these cells.
Just know what they say.
Yeah, yeah. If you know that, you're fine. And just kind of know that they're beginning to pull information together to send down our neurons to our brain. Is everyone good? All right, so basically you're seeing, this is just a quick synapse of what they do. Our horizontal and our Maestrin cells, they do communication between photoreceptors and ganglion cells. The big thing to know about these guys is they alter the sensitivity of the retina, which is important because if we're in a really bright outside setting and the light's really almost blindingly bright, these guys have the ability to like tone it down and make your retina less sensitive to light, which, and I think I kind of mentioned this last time. So if you're outside and it's really, really bright and then you come inside and it's dark, you kind of can't see at first. And it's because these guys basically turn the dial down and said too much light, too much sensitivity, and they'll tone it down. Can they adjust back? Yes, absolutely they can. Same thing if you're in a really dark room, you get where you can kind of see stuff and then you kind of adjust to it. So it's altering that sensitivity. Of course, it's also using the size of the pupil to regulate light coming in. Optic disc, we talked about. Everybody good with the optic disc? Chambers of the eye, we talked about. Everybody good with the chambers? Just know your anterior is your small one. It's in front of the lens, right? And your posterior. And just, I want to show a picture just because on this picture right here, there is a tiny bit of space between the lens and this area here. This is all still posterior chamber. So kind of just the very top of the lens and back is posterior. Everything kind of from the pupil or the shelf that holds the lens up into that into the center point of the pupil is going to be anterior. So it's not like the lens pushes tight up here and totally separates that. You just have a little bit of posterior chamber that's slightly in front of the lens. Let's see, we talked about aqueous humor. It's circulating and it goes through the anterior and the posterior cavities. Interocular pressure, this is basically how full our eye is. It helps to maintain that shape. And this is what they check if you ever have where they blow the little puff of eye or of air on your eye. That's what they're testing here is your interocular pressure. If we get too little of pressure or too much pressure, it's going to affect our vision and could do permanent damage to the eye. And I'm going to go to the picture one more time. Is everybody good? So if I go to this picture here, you can see right here where I have my aqueous humor can really circulate into all these areas. So it really travels from anterior to posterior, but our posterior holds the vitreous humor. The vitreous humor is not going to be up in the anterior chamber. Aqueous humor really can travel between both. Why? Because the lens, it's not like it's pushed up here sealing off the anterior chamber. All right, and the other thing I want to show you is a ciliary process with these ligaments that attach to the lens. And as these ciliary bodies contract or relax, we can alter the shape of the lens. So they attach, these muscles right here attach through these ligaments to the lens. Okay, the lens is held in place by the ciliary zonule, and I just kind of showed that to you right here. That's these guys right here. What should you know about the lens? You should know that it is made of a protein called crystalline, which is a very, very crystal clear protein. It's crystal, that really clear, lucid part of it is what makes it where we can see. If it gets really foggy, then that's what a cataract is. And we kind of talked about that. You lose, like on the cows, we could have said, oh, they all have bad cataracts. They've actually denatured because they were dead. Probably when they were alive, they were crystal clear on most of them. But you'll see animals will get these old age cataracts. Basically, if you've ever had a really old dog and you look in their eyes and they're super cloudy in their eyes, that's what your cataract is. And it's basically your lens just losing that transparency. Okay, all about focusing is how we bend the light, and we've really talked about that. We're trying to bend it to our focal point. What's our focal point in our eye? Our fovea centralis. And we're going to have a focal distance. This is the distance between the center of the lens to the focal point. So basically, what we're looking at here to the lens and then back to our fovea centralis. And I don't ask a lot of details about this, so don't get caught up feeling like you have to spend a ton of time studying this. What I really want you to understand is who does the light bending, where are we trying to bend the light to, and what happens when we alter the shape of the cornea and we alter the shape of the lens. You don't have to know all these little details of this. What you should know is the rounder the lens, the shorter the focal point. This is where I'm aiming on my fovea centralis, is my focal point back here. So round lenses give you a shorter focal point. A couple terms you should know this. Astigmatism, I already talked about this. What did we say it was? Yeah, misshapen cornea. It's where I do the little football kind of cornea. So when your cornea gets misshapen, it is the structure that does the most amount of light bending. So if it gets misshapen, it's going to bend the light into the wrong part of the eye. It's not going to hit that fovea centralis or the focal point in the back of the eye that it should. It very often mimics nearsightedness. Accommodation, this basically means I can see clear far, I can see clear close up. So your eye, your lens makes those adjustments to allow you to focus at all kinds of different distances and when things are at different locations close to your eyes. And again, our lens gets rounder to focus nearby. It elongates to focus farther away. That's really what you need to know. And what happens when we get older is our proteins get a little stiffer. They don't allow that lens to make those adjustments as well. And we can't contract and make a nice round lens as easily as we used to be able to. And that's when people start really needing reading glasses. It's just a sign that your crystalline in your eye is getting stiffer. And this shows the rounding of the lens here. Our ciliary muscles are contracted, which rounds the lens. Here's our focal point back here at our fovea centralis. So you can see we bend the light here. We bend the light by adjusting the lens, and we get all those light rays to focus on the focal point.
That'd be close up.
Yeah, this would be more of a close-up because we have a rounded lens. And then you can see, so here was my starting point. So I was pretty close to the front of the eye. Now when my lens flattens, my starting point's clear out here somewhere. So the farther away you're starting to look, so this is your distance vision, we're going to use a flatter lens. And we're still going to bend that light to the fovea centralis. I always think it's kind of counterintuitive. It feels like because these guys are kind of at the top and the bottom, we would contract to elongate, but it actually contracts to round those lenses. 20/20 is the vision that we're all looking for, which I'm sure you guys know. Scotoma, just know the term. It just means you have an abnormal permanent blind spot. What can cause blind spots? Lots of different things. Injury, it can be compression of the optic nerve, damage to your photoreceptors. It can be central damage to the iris, the cornea, the lens, any of those kinds of things. When you, and I think I kind of talked about this last time, when you see, when things come into your eye, they actually are upside down and backwards when they focus into the back of your eye, and your brain turns them around and makes sense of them. And remember last time I was talking about, they did the study where they put the glasses on the person and everything was upside down, and the brain ended up flipping everything around. And then when they quit wearing the glasses, they had to readjust and flip everything around again. So we see upside down and backwards, but our brain adjusts everything from your eye. This is, so if you were looking, something here is actually hitting down here. So this is just kind of showing you it's the upside down point, and this end of the object would end up being more at this point up here, like that. So they flip upside down. And they flip backwards. So on this one, the closest to the eye is the pink. Back here, it's the farthest from the eye. And then, like I said, your eye turns it around. All right. Myopia is nearsightedness. Just know that term. And what does it mean? It means that we have too deep of an eyeball when we have myopia, and so we end up with this focal point that's kind of in front of that real focal point. We're hitting a focal point that's too close. And that's the most common, right? People can see close, but they can't see far away. So we get corrective lenses so we can see better far away. And then hyperopia is farsightedness, where we can see fine far away, but we cannot see very easily up close. So this is where our focal point's too far back. So they're not hitting our fovea centralis where we want them to be, because we're either aiming short of it or beyond it with our refraction. Yes. That's a great question. I don't actually know the answer to that. I think that's more of a processing thing in your brain, but so I think it might be related to that, but I'm speculating. I don't honestly know the answer to that. That's a great question. Okay, and then what is PRK or laser eye surgery? Basically, they go reshape the cornea. Because if we can fix the front of the shape of the cornea, we can bend the light to where we want it to go. It'll fix your vision. I've had it twice. I have super bad astigmatism, and it just doesn't last. It lasts about 12 years, and then back in glasses again. I don't know if I want to pay for it again. It's nice, but it's expensive.
You just have to wear glasses instead.
Yeah. I'm not near as blind. The first time I got it, I had like Coke bottle glasses. I wear contacts all the time, and I started having a lot of issues where I couldn't wear contacts, so I got it. And it did last me about 12, 13 years. It was awesome. And then they've slowly gotten worse, but they're nothing like what they were before. So I've had one eye done 3 times and the other eye done twice. I think my right eye is my really bad eye. What's that? It was like $2,000 an eye.
Is it like cosmetic where it's like?
Yeah, your insurance won't pay for it, which stinks. I know, because the first time I had it done, like they could no longer correct me to 20/20 vision because it took so much refraction to do it. So I was like, I feel like this should be medical, but
Yeah.
they won't pay for it. So, all right. You guys don't need to worry about this information. We're just gonna skip through that. All I really care about you knowing on this is that retinol is synthesized from vitamin A. So we do need vitamin A for good, healthy eyes.
What's retinol?
It's a protein that I don't even know. It just contributes. Yeah. Well, it comes from vitamin A, but it just like in skin, it gives more collagen, more flexibility, more resiliency. This is going to be more of a protein, but not really. I mean, those are a lot of collagen proteins. These are,
I know there are like different ones. There's
Yeah.
retinol and then there's like retinoid.
yeah. All right, and this just is a blow up of what we were seeing before. Here comes our light. It's going to go all the way through to the pigmented layer of the retina, and then it's going to come forward. It's either going to hit our rods or our cones. These are going to be our color. These are going to be just our big light sensitive receptors. Color vision, you have blue cones, green cones, red cones. If you are missing any of those cones, which is a genetic trait that you inherit from your parents, then you will not be able to see those colors. Each of them have a different opsin, which is a protein also, so. You are just missing those specific color cones. This kind of just shows you the wavelength of those. So you're not going to read those wavelengths of light. They are the receptors for those light waves. Here's a little test. Can you guys all see that? We don't have any guys in here. Usually it's guys that are colorblind. Why guys instead of girls? Do you guys know? It is genetics.
I don't know what.
But it's because it's carried, it's linked to the X chromosome. And girls have two X chromosomes. So even if you inherit color blindness from one parent, not being colorblind is a dominant trait. So your other one will cover it up. So you would have to inherit a color blindness gene from both parents for a girl to be colorblind. It happens, it's not very rare. But guys, because they're X and Y, they are only inheriting an X from their mom, a Y from their dad. So if mom passes them an X colorblind gene, they're going to be colorblind. So they don't get another one to maybe cover it up. So if we're in a dark adapted state, that just, we kind of already talked about this. This means that you're in a really dark room. It's going to make us where we're going to see those lights a little bit better. And visually, our pigments get more stimulated, so we see a little better. In a light adaptive state, we're going to basically tone it down. Retinitis pigmentosa, this is an inherited disease. This is just retinal degeneration. So our retina slowly begins to degenerate. We cannot see without our retina. Why? It's our neural layer. It has those rods, it has those cones. If you get tears, damage, degeneration, you're not going to be able to see. All right, so basically visual pathways, it begins at the photoreceptors. That's going to be out at those rod area, and then it's going to end on the cerebral hemisphere. So we start in the photoreceptors in the eye, and we're going to end up in our cerebrum. What area of our cerebrum receives our visual information? It is occipital. And we also receive some of our visual information into our superior colliculi, but that's not part of our cerebrum, right? That's more in like midbrain area. All right, your M cells monitor rods. These guys do general information. So what do rods do? They're doing general information, motion, shadows. They're not our crystal clear vision, right? Who does our real detailed crystal clear information? Reception, it's our cones. So M cells are monitoring rods. So they're doing general information, general vision, nothing clear. That's all you need to know about that. And then our P cells, these monitor the cones. And this is where we get our fine detail in our color. So M cells are rods, real general vision. P cells are cones. This is going to be more of your detailed color vision. You don't have to worry about that. Optic chiasm, you guys all know what that is, right? So our ganglion cells go to the optic disc. They come in through the back of the eye to the optic chiasm. And then they're going to go to the visual cortex, which we said is occipital lobe, and we may have some of that reflexive information go into the superior colliculi. You do not have to know any of the nuclei for the eye vision or the eye stuff, neural transmission. Circadian rhythms, you should know that. What is our circadian rhythms? Day and night. And when we look at even the seasons, like if the sun comes up, at least for me, unless you are artificially blacking your room, when the sun kind of rises and your room gets light, it wakes you up. It is part of that circadian rhythm that you see through your visual processing. And this is why, well, and it affects metabolic rate, blood pressure, but it affects mood too. It is true in the winter where we have less light, it's affecting the circadian rhythms. And that's why people get like more depressed moods and things. So kind of those winter blues is a real thing. And it has to do with the visual light processing and the rhythms. And this just kind of shows, you can see that some parts of our eye are doing kind of one extreme or the other, but we also have parts of our eyes where both eyes are working together to see it. You don't have to know any of these pathways. The big thing is, and you don't even have to know this, I just want to point it out to you, that we're going to come into the diencephalon of the brainstem, and then we're going to eventually end up here on the occipital lobe. If needed, if it's a reflex, it's going to come into that superior colliculi, which is at our brainstem, midbrain area, right? You guys remember that? Okay, let's go to the ear. I like the ear. I think the ear is super interesting. You have three big sections to your ear. You have your external ear, your middle ear, your internal ear. What is the big division line between external and middle ear? It's your eardrum, called your tympanic membrane. So your eardrum splits your external ear into your middle ear. Once we have our three little bones in our middle ear, and once those three little bones kind of bang on another membrane, we get into our inner ear structures. So let me show you a picture. Our ear is going to be doing our hearing and our balance, and we're going to look at those kind of separately and how they work. So here's my external ear. This is the auricle. Here would be my ear canal, or my external acoustic or auditory meatus. You guys remember that from 201. Here's my eardrum, my tympanic membrane. What happens with hearing is we have these sound waves, and they really come in waves, and depending on the pitch of the sound, the decibels of the sound, they're going to come in closer together, or they may come farther apart. But either way, as that sound funnels in, it is going to begin to shake the tympanic membrane. As it shakes the tympanic membrane, we're going to see it go to our three auditory ossicles, which are our smallest bones in our body. So the membrane wiggles this one, which bangs on this one, which wiggles this one, which bangs on a little window right here. So that's how we pass our sound from our external ear. This area right here is my middle ear. And as soon as I bang on this membrane here, this is going to be my inner ear area. You should know this. This little drainage right here is called the auditory tube, or the eustachian tube. Have you guys heard of that before? With kids that get a lot of ear infections, what happens is sometimes they don't have a proper opening to this auditory tube, or it's not mature enough, big enough to drain down into you should know this, it drains into the nasopharynx. So your auditory tube drains down into your It's draining inner ear fluid or middle ear fluid. I shouldn't say inner ear, but middle ear fluid. So when you get an ear infection, you may build up, like maybe get a bad head cold. What happens is you're going to build up some fluid here in your middle ear. And if it can't drain here, it's going to build, and it begins to bulge your eardrum out, which is going to cause a lot of pain. We have a lot of neural receptors to our membrane. Why? Because we want to protect this eardrum membrane. Because if we break it, are we going to hear anything? No, because it's not going to be there to wiggle these bones to move our sound around. So we have a lot of neural receptors in here. So as this membrane bulges out, it gets sore, it gets painful, and sometimes it'll get so bad it'll break from the inner fluid pushing out on it. And once that happens, it will heal again, but it can create a lot of scar tissue, which affects the elasticity, the movement of that eardrum. So kids with chronic ear infections, and if they've ruptured their eardrums, it can affect their ability to hear because we build up a lot of scar tissue and things like that. So sometimes I'll go put in what they call ear tubes, and they look like these wee little baby straws, like a little section of straw, and they'll actually do one little opening and put that little straw-like thing right through the membrane. What does it do? It allows fluid to actually drain out through the external ear to keep this middle ear, because it's much better to heal one little teeny hole than to have it rupture and blow the whole eardrum out. So sometimes I'll put a little auditory tube in there till kids develop a better eustachian tube. And they actually just, as the membrane heals, it'll push the auditory tube out. All right, so what we're going to be looking at is we're going to dig into this area, the auditory ossicles. We basically take sound wave movement, we turn it into this mechanical movement. So we're going to mechanically move this, which is going to mechanically move these three little bones. These little bones are going to bang on a membrane over here, and inside these structures over here is a bunch of fluid. And this banging on this is going to begin to create movement of my fluid. And then we have our little receptor cells, kind of like we've seen with the other specialty senses. We have our neural receptor cells that are going to receive signals based on how the fluid gets moved inside of these areas. So that's kind of where we're headed. This up here is my balance area. And this down here is the cochlea. This is the hearing area. And you can see this is my vestibular cochlear nerve. It comes in and it splits into the vestibular nerve, which is doing my balance, and the cochlea, which is doing my hearing. So this one nerve is going to branch into, one's doing hearing, one's doing balance. So we'll be looking at each of these structures individually, this one and this one. Okay, so we talked about what the external ear is, the auricle. The tympanic membrane, your eardrum. Is everybody good with that? If I kind of truck on, it separates external and middle ear. Do you guys need a minute, or is everybody okay? Slow me down if you need time. All right, you have a ceruminous gland. This basically is secreting your earwax. Why do we secrete that? Well, it keeps things from hitting against your eardrum. So you get a bug come in your ear, or you get dirt come in your ear. When it just protects that eardrum from getting damaged. It also has enzymes in it, which is going to slow the growth of microorganisms, which is pretty easy to grow in your ear. Honestly, if you are a swimmer, you're swimming in lakes, have your ears in the water a lot, that wax can help to kind of hold that water in there, and then it's a great environment for stuff to grow, bacteria and things like that. That's what swimmer's ear is. If you spend a lot of time in the water, they get a lot of water in their external ears, and then it just becomes this great environment for bacteria to grow. So you basically get external ear canal infections. So usually something antibiotic will help. Okay, so our middle ear starts at our eardrum. It's called the tympanic cavity. That is our middle ear. It's an air chamber. And I already talked about, we have that eustachian tube or auditory tube. that drains down into the nasopharynx. The other thing it does is it helps some with sound. It's going to resonate some of that movement, that pressure down into that tube, because we build a lot of pressure with all that movement and those sound waves coming in. And so your auditory tube, not only can we drain fluid from infections, but we also can drain the pressure from all that movement in there. And that's why when you're like driving or flying and your eardrums get plugged and you hold your nose and then you blow, you're basically opening up those auditory tubes to equalize the pressure in your ears. Your auditory ossicles, there's the three. We have the malleolus. That's the first one that the tympanic membrane wiggles, is the malleolus. It's also called the hammer. The incus is the anvil. So the hammer hits the anvil hits the stapes, which is the stirrup. So this is the first one that wiggles. It hits this one, which is the second one, which hits this one, which is the third one. And the stapes is actually going to hit something called the oval window. And when it hits the oval window, it begins to move that fluid. And the waves of fluid are going to move our receptor cells. They're going to be mechanically gated ion channels. How do we say mechanically gated ion channels work? Yeah, we move something, right? We distort something, which opens up those channels. So that's the fluid moving is going to cause a movement that opens up my ion channels. All right, I'm just going to backtrack to my picture real quick. Here's my malleolus. Here's my incus, and here's my stapes. It looks like a stirrup, right? Okay, oh, the next picture was that. I didn't realize that. All right, so here's my eardrum, malleolus, incus, stapes. We have some muscles in our middle ear that can also make our hearing a little more sensitive or kind of get it to tone it down. And that's that tensor tympani or stapedius. And these will control the ability of the malleolus and the stapes to wiggle. So if you're at a rock concert and it's really loud, have you ever had something so loud like your ears just kind of ring? These muscles are going to flex and make it where these don't move as much, which is going to help to tone some of the sound down. So that's what they do. If it's a real quiet environment, they're going to let them move and flex more. All right, and this just shows you a visual of those. This shows these guys individually. All right, so I already talked about these. The tensor tympani and the stapedius is going to reduce movement. If we reduce movement, we're going to reduce what we hear, right? We're going to desensitize our hearing a little bit. And this is all designed to kind of protect the ear from very loud noises. Is everybody good? All right, this is where we get into the interesting stuff, is we get into the internal ear. In our internal ear, we have our fluids, as I talked about. We have a bony labyrinth, and we have a membranous labyrinth. So we would basically have bone, and then we have a membrane, and we have fluid inside the membrane, and we have fluid between the bone and the membrane like that. So this is bone. Here's my membrane. And we're going to have fluid on both sides of this membrane right here within the bone. The perilymph flows here. So this is going to be perilymph. This in here is going to be my endolymph. This is peri, and this is endo. Just think of endo is in. Endo is in.
I like a periosteum.
Yeah, exactly. You got it. We can divide our bony labyrinth into a vestibule, semicircular canals, and the cochlea. This would include our balance stuff and our hearing stuff. The vestibule and the semicircular canals, these are my balance portions to my bones. The cochlea is my hearing portion. The vestibule does vertical balance and horizontal. So if you've ever done the rides like at the carnivals where they haul you up and you're sitting there and all of a sudden they pull the thing and you drop, this would be the vestibule that's detecting that fast up, fast down. It also detects fast forward, fast back. Your semicircular canals do more orientation type positions. And then your cochlea is all your hearing apparatus. All right, so let's dig into the two of these. This is what they look like. Here's my cochlea. We're going to come back to this. This is all my hearing apparatus, the little snail thing. All the rest of this is my balance. The vestibule portion is down in here. And then we have our semicircular canals right here. What you can see on this picture is here's our bony lymph, or I'm sorry, the bony portion. And then here's my membranous portion on the inside of the bony portion. All right, and it would look like this. This is kind of what I drew on the board for you. Here's my bone. Then I have my perilymph between my bone and my membrane. And then inside the membrane is endolymph. All right, we have something called the vestibule. This has the saccule and the utricle. And these are the ones I was saying do forward and back, up and down. These are doing those vertical and horizontal movements. And then our three semicircular canals are going to be doing rotational type movements. And they do it based on the rotation of the head. But if my whole body's rotating along with my head, even this kind of stuff, I'm going to detect that. And then our cochlea, which is going to do hearing. And we're going to look at each of these three things individually. We're going to start up here with the vestibule and the semicircular canals, and we'll do the hearing of the cochlea last. All right, in the internal ear, we have, at least I thought that's what we did. Yeah. All right, in the internal ear for the hearing portion, we have the round window and the oval window. So when the stapes bangs on the membrane, it bangs on the oval window first. And remember I said it moves fluid? Basically, if I took my little snail like this, it bangs on this and we're going to see fluid go all the way up and come all the way down because it's almost like two little passages. So the stapes bangs on one window. The window that it bangs on is the oval window, which starts the signal going up here. And then it's going to cross over, do the hearing, which I'll talk about how it does that. And then it's a physical moving thing, so we can't really trap it in there. We have to let that pressure come out. And so when it comes back down, it's going to come out this way. It comes out the round window. So this is leaving pressure. And this is incoming. And what creates this pressure, this wave pressure, is the stapes, the banging of the stapes. I always remember the round window is where it comes out because I think of you round it out. If we're going to round something out, that's how I remember. All right, equilibrium. So we're going to go back to the equilibrium. Basically, what is equilibrium? It's kind of knowing your body position, knowing your orientation of your body. How do we do that? This is all called the vestibule and the semicircular canals. And basically, we have little hair receptors. So we're going to have these different hair receptors that are attached to an area with some neurons. And I'm just going to draw some kind of silly looking ones here. These are mechanically gated ion channels. So as my fluid flows one way or the other, it's going to bend these hairs. As the fluid hits it and bends it, that is going to cause my mechanically gated ion channels to open And then I'd have a nerve attached to this, and then I'm gonna see an action potential going down my nerve. What triggered that action potential go down my nerve? A mechanical movement that distorted the membrane and sent an action potential. And I'll show you some pictures. All right, on our semicircular canals, we have some that are doing this direction, and we have some that are kind of going more at an angular direction. So we have something called the utricle. We have something called an ampulla, an ampullary, cupula. Do you guys know what a cupula is, like on a barn? Have you ever seen a barn or a building like this, and they have the cute little box on the top? It looks like a little house sitting on the top, and it might have a vent or some little cute thing on top of it. This is called the cupula. So I always think of, you're going to have like a little house up there on the top is a cupula. What we see in here is our cupula is made up of gel-like stuff. So this is a gel, and it's going to have, let me write this where you can read it. And inside this gel is going to be these specialty hair cells. For the most part, there's one that's different. But as the fluid pushes the gel is basically going to slosh to one side or slosh to the other side. And as the gel sloshes, it moves the hairs. As it moves the hairs, it opens up my gated ion channels and sends my action potential. And I'll show you a picture. I think when you see the picture, it makes a ton of sense. All right, so our gel, our little house on the top, is our cupula. And it has the hair cells that bend. Those are called the ampullary crest. And the individual hairs can be called stereocilia. And it has one large one, which I kind of drew here. You have one large one, and then the rest are smaller. This large one's kinocilium. And they actually look a lot like microvilli. They're just like little finger-like projections.
Sorry, what's
Kinocilium right here. So you have a whole bunch of these little guys and one big long kinocilium. These are in my semicircular canals. Okay, so here's a picture of these. Here is my semicircular canals, and what's going to happen is as you tip your head one way or another, or your whole body tips and your head goes too, it makes this fluid in here begin to turn in these semicircular canals. So this is all fluid moving. This is my little ampulla, and it has the cupula in it with my little hairs in the center. So this whole gelatinous mass, when the fluid goes in the semicircular canals, it's going to push these over, which is going to move the hair. So let's look at a picture. This is a blow up of this right here. This one's turned sideways. When we look at the picture, it's straight up and down. So this is straight up and down. Here's my little cupula, like the little house on the top. Here's my little hairs right here. And here's my fluid in my semicircular canals. So here's our fluid. Here's our little cupula right here with the in the ampulla. So the ampullas are kind of these opening areas of these semicircular. Okay, so as fluid moves around the circles based on our movement, it's going to push this whole gelatinous mass to the side. When it does this, it's going to bend these hairs, and then we're going to get that action potential. You can see these hairs, it's going to be mechanically gated ion channels, which are going to send an action potential. Why? Because they open up those sodium ion channels, and then it's going to send my action potential right down my sensory neurons. And this shows kind of, here comes our rotational movement in our semicircular canals. And if my fluid was moving this way, it bends the little gelatinous mass, it bends the hairs, opens up my sodium ion channels, and boom, I have an action potential go. How do I know what that direction is? Well, it depends on which fluid moves. One semicircular canal does this kind of circular movement. One does this kind of circular movement, and then we have one that's kind of more of an angular one.
But do they all have the same result?
They do if the fluid is moving in the direction that they detect. So if I was saying doing a forward roll where I'm rolling all the way over, it's the semicircular canal that detects this. The others would detect nothing.
Oh,
Does that make sense?
yeah.
So if the fluid gets to moving in the direction you're going, those are the ones that are going to bend and send the sensory. The others will be unaffected because it's not going to be in that plane that creates that fluid to move around the semicircular canals. So that kind of shows you how that works. We use the endolymph, so the fluid that's inside of here comes along, pushes our little cupula of gel like this. As the gel moves because of the fluid, it bends the hairs, which sends the action potential. Yes.
We spin around them all.
Yeah.
Why does that stomach?
Oh, I don't know why stomach. It's just part of the vertigo, but I don't know why. But when you're still spinning and you still feel like even when you stop, it's because your fluid's still moving in your semicircular canals. When you've done a lot of spinning, you kind of get that fluid going like that. I don't know why it affects your stomach. All right, so this shows it here. Here's my little Gillette. This is just a zoom in of the hairs. So here's my gel. As it moves, it's going to bend these hairs. This is a cell right here, which we're going to see sodium ion channels. We're going to see action potential that's going to go right down my sensory neuron then. All right, utricle and saccule, these guys are the ones I was saying do up, down, forward, backwards. They do those linear motions. My semicircular canals are going to do more of those circular. These guys are going to do linear motions. And they are slightly different in that they still have a gelatinous mass, but they look a little different on the inside of them. So let me show you those. Are you guys good? All right, you have basically an area where you have a cluster of hair cells. I always think of it like a little mat. We're gonna have a little mat with some hair cells. And we're also gonna have a bunch of little stones in there. They're little calcium stones in there. And they are called otoliths. They call them ear stones. So in this little mat with my hairs, I have a bunch of little crystals. And if we were accelerating and then we slammed on the brakes, what would happen if we had something in our car? It would go forward, it would keep moving. This is how these work. As we're doing a linear movement and we take off, all these stones and everything will displace to the back and triggers the hairs that's in the action potential. If I did like a quick stop or something, all my little stones would go here. If I tipped my head, all my stones would go to one direction and it would trigger that sensory nerve that, hey, the head's done a linear movement. So that, let me show you a picture. I think it makes a lot of sense. The utricle does my horizontal movements, that's frontwards, backwards. The saccule does my up and down movements. Is everybody good? All right, so this shows our utricle, this does our saccule. So they're still big chambers. They're just at a little bit slightly different location in this whole labyrinth here. All right, so this is showing the utricle, and this is my little gelatinous mass. Here's all my little crystals. So if I swash everything to one side, do you see how I'm going to bend hairs? And they're going to be attached to different sensory nerves. And those are going to open up sodium ion channels and just send my signal down the ones that are bent. These guys are going to be detecting nothing. Why? Because they're not bending. And this helps me to know, based on which nerves get bent and send an action potential, what that location is. So we have, again, just this big gelatinous, I always think of it like a little mat right here with your stones. And as you tip, the gel tips and the crystals tip, and that's what caused these hairs to bend and send an action potential. And interesting, I've heard that people who get a lot of like vertigo and things like that, sometimes their otoliths are out of place. And there's some sort of like manipulative movement you can do that supposedly puts your otoliths back in place. Or so I've been told. I've never experienced it, but I thought that was kind of interesting. All right, so this shows an example. His head is normal. His little mat is straight. All of his crystals are just sitting straight down. Nothing's bending. As soon as he tips his head, the gel all sloshes to that end and the crystals all slosh to that end, and they bend these hairs and they send the action potential. They're using mechanically gated ion channels. These are just going to go, really all you need to know is this is going to go to the vestibular nerve, which is going to be part of the vestibular cochlear nerve. So vestibular nerve comes from the vestibule, cochlear nerve or auditory nerves comes from the cochlea. They join and become the vestibular cochlear nerve. I won't ask you about the nuclei. And this, again, is cranial nerve number 8. But you guys know that, right? Because we did cranial nerves.
What do you want us to know about it?
I just want you to know that the vestibular nerve comes from the vestibule. The cochlear or auditory nerve comes from the cochlea, and they join and become one nerve as they enter into the cranium, and it is the vestibular cochlear nerve. That's all you have to know is one's going to come from one structure of the inner ear, one comes from the other structure, but they do become a common nerve. And I'm not going to quiz you on this nuclei, so don't worry about that. But this shows your vestibule. So here's my semicircular canals. We know they're bending by moving the fluid, right, in the canals. And then our utricle and saccule are going to be doing those up, down, forward, back movements. As they fire, these nerves come in on the vestibular nerve, and then this would be my cochlear nerve. They become a common nerve. Out here, the vestibular cochlear nerve. All right, some autonomic movements of the eye. I don't know why this is right in here. It seems like this is out of place. I don't know if my thing got jumped or not. Did we talk about this? Weird. All right, nystagmus is basically, this is what they do on roadside tests is they'll test for nystagmus when they think you're drunk and they have you follow their light with their eye. What happens is when you get all the way over here, your eye will sit there and bounce and move and you can't control it. When you've had alcohol in your system, that is called nystagmus. It is like involuntary eye movement. It can be damage to the brainstem or internal ear. I guess this is why we had it with the ears, because it can be damage to the internal ear. But it is also caused by drinking too much, which would affect portions of our brainstem. But temporarily, right? All right, so for hearing, we're going to go in. I already kind of talked about this. We're going to take sound waves. We're going to turn those into mechanical movements, which are going to create pressure waves on our fluid. And that's how we're going to get our sound. So let's look at these. Just a term you should know, frequency is the number of waves per cycle. Hertz is the number of cycles per second. So just kind of know those definitions. That's all you need to worry about. Pitch is our sensory response. Amplitude is the height of the sound wave. And then intensity is the amount of energy, which basically is our decibels. When we're giving a decibel number, this is what we're doing the amount of energy in a sound wave. So basically just understand those definitions. If you have the definitions down, you're good. Is everybody good? All right, so this shows basically gives you an idea of the wavelengths, and this is how that sound is going to come into the ear. And it's going to move that tympanic membrane kind of based on the energy from each of these waves. And this is just kind of interesting. You don't have to know anything off of this, but you can kind of see the decibel levels of some various things. Some really loud ones, traffic noise, heavy metal rock concert. Okay, so how do we turn this movement into a sound? We have the cochlear duct, and we have the scala vestibule, or the vestibular duct, the scala tympani, which is the tympanic duct, and we have the organ of Corti, and a basilar membrane. All right, so how does this work? If I took my little cochlea, which is my snail, and basically it's a double coiled tube thing. So when I go up my cochlea, let me draw this better. It's like I'm going to take two tubes right next to each other, and I'm going to do this double coil of these two tubes. One of the sound comes in. Which window does it come in? Oval. And it comes out, which creates this is the stapes. And it comes out the round window. Okay, if I unwrapped my cochlea and I turned it into just two of my tubes next to each other, so I'm just going to unroll this big snail shape here. This is basically what it looks like uncoiled. And here comes my incoming one. I'm actually going to do it up here. So this is my oval window, and this is my round window. My sound waves are going to come out this. So they would go some amount up my coil. They're going to cross over this membrane and come down. How do the sound waves, which turned into fluid, because these are full of fluid, this is called my vestibule, the part where it comes in, the big open part where it comes in is the vestibule. So when my fluid comes in, this right here is called my basilar membrane. And it will cross across the basilar membrane to the part where it's going to come out. That membrane has different amounts of flexibility to it. And the amounts of flexibility to it determine where it will cross over. Some sounds will cross over this close, so they'll come in, they'll go out. Some sounds will travel all the way up that little snail shape, and they may not cross over till here, and then they'll come out. It is based on the flexibility of the basilar membrane. It has all these little areas of flexibility, and based on the sound, it will allow it to flex at that location, and then it moves the fluid as it comes around. This part down here is the scala tympani. All right. So what happens, where all the magic to hearing happens, is right here kind of at this crossover point. And let me jump to, I'll just show you this picture. Here's my stapes. Oh, they put theirs on the bottom. I was trying to remember which way they did theirs. But this would be my vestibule. This would be which window? Oval. And they're going to go up. It's going to spiral up. At some point, it's going to cross over and go back down through the tympani. And it's going to come out the round window. What is it? Pressure. The pressure has to come out. We can't just dissipate it in an enclosed space. So we have to let the pressure back out. Okay, and this shows it coiled, which I think is difficult to see. So we're going to look at it kind of this direction. This would be one area. So if I took one area right here, this is what I would be looking at. I'd be looking at my vestibule, the area that has my basilar membrane, and then my tympany. So here's my vestibule right here. This is where my fluid would be moving going up the spiral. What happens is as the fluid goes up the spiral, it begins to vibrate. It passes that vibration into the middle part. And this is my basilar membrane. And as it does that, it begins to wiggle this basilar membrane right here. So as the fluid moves, if this is flexible, it moves the pressure over here and it begins to wiggle this basilar membrane based on the pressure, based on the flexibility. Remember I said different sounds have different flexibility in different locations. So wherever the location is where it allows it to be flexible, we're going to see it begin to wiggle this basilar membrane. You can see we kind of have this thick part right here, and then we have kind of a roof to the thick part. Our little receptor cells are in this part right here, and this little plate sits above these cells. It is called the tectorial membrane. And what happens as the basilar membrane begins to vibrate up and down, it bangs these cells against the tectorial. So it's kind of like I have a roof, and then all of a sudden I start wiggling everything, and they all start hitting their heads on the roof. That's kind of how we bend our hairs here. So here's my tectorial membrane sticking up right here. Here's my basilar membrane with my little receptor hairs. As this begins to vibrate up and down, it begins to bang these guys, bang their heads on this membrane. As it bangs its heads, it's going to bend those hairs. So it's going to do mechanically gated ion channels. It's going to open up, and they're going to go down the nerves. The pressure is going to cross all the way across into the other big chamber here. So this is my incoming chamber. If I hit the flexible location, it begins to wiggle this basilar membrane, which causes these cells to go up and down, bang their head on the tectorial membrane. Now, boom, that sends a synapse down this nerve, and my pressure goes into this other chamber and exits through the round window. So we're basically passing that pressure in the wave of fluid from this to the wiggling of the basilar membrane and then down into the scala tympani. Does that make sense? Here's a zoom in of it. Here would be where my fluid's coming through. If it was at that flexible location, we'd see this begin to vibrate. These are my receptor hairs. They're going to bang their head on the tectorial membrane, and I'm going to see my action potential go. And then my pressure is going to move over into the scala tympani, and it's going to move its way down the little snail and out the round window. And this is a zoom in of those little heads on my basilar membrane. So there are these cells with the little hairs. As it begins to wiggle up and down, again, these little hairs are going to bang their heads on here. When they do that, they bend the hairs over, open up our sodium ion channels. Boom, we get an action potential right down our nerve fiber. Then where's our pressure from our wiggling going to go? Into the fluid down here on the other side of the basilar membrane. All right, so how do we discriminate between sound? It depends on where the flexible location is on my basilar membrane. Not all sounds are created equal. Like I said, some tones of sound will have flexibility here. Some tones of sound will be way up high in the snail. So how do we determine it? It just depends on where flexibility is. With age, we damage those particular things. We tend to hear our highest pitch sounds down here, lower pitch sounds farther up the spiral. So what's the first sound that tends to go with older people? Those really high pitched sounds. Did you ever do a little buzzing test? I know your phones can do it, and some people in this room are like, oh my gosh, and other people don't even hear it at all. That's right here on these. But as people get older, these get damaged. Why? Because they sustain a lot of wear and tear. and you lose flexibility to that membrane. And now we don't even hear those. We travel up and we hear those louder sounds. So as our membrane gets stiffer and less flexible, we're going to lose that. What's that?
I'm saying that you stop hearing all the other stuff.
I know, kind of, I guess, but it is frustrating sometimes, probably,
Well,
I
yeah.
know. I know,
Or
older
sometimes
people,
those things you can't hear, like a whistle.
I know, exactly. Some stuff you're like, eh. And any form of mom, mom, mom,
Yeah.
mom, you can't hear that anymore. right?
See, that's how it's happened.
Exactly. Another thing that causes hearing loss is our ossicles get stiff. They can get arthritis and not wiggle the way that they should, just like other bones can. They're still bones. They can get stiffening and arthritis between those bones, which is going to affect how much they bang on our oval windows. So it's going to move our fluid less, and we're not going to hear it.
You get that diagnosed.
Oh, I've never heard it diagnosed, but they probably do. It's kind of crazy how that I don't even know how they do that. Like cochlear implants, I don't even know exactly how they work, but somehow they magnify in the cochlea. Does anybody happen to know how they work? If anybody, sometimes people have family that have cochlea and they know how they work. But my understanding is they put the receptors right in here to the cochlea. They put a little implant right here and it goes right into the cochlea. And I don't know. I just think like science is amazing, the medicine they've come up with now.
People that have tinnitus, that have ears, is that the
I don't know exactly. I would assume because it tends to be high pitch, it's got something to do down here, but I don't actually even know exactly what causes that. All right, so 6 basic steps of processing. We've already talked about this, but we'll just look at it. Sound wave arrives at the tympanic membrane. What's our tympanic membrane? Eardrum. Boom, boom, boom. We're wiggling our eardrum. What's that going to do? It's going to move the first ossicle. What's the first ossicle? Yeah, malleolus. It's a hammer. What's the hammer hit? The anvil, which is the incus, which wiggles the stirrup, which is the stapes. What's the stapes do? Yes, it bangs on the window. Which window? Oval window. And now we turn it into a fluid movement. So here comes our sound waves. Wiggles our eardrum, wiggles the malleolus, wiggles the incus, wiggles the stapes, which bangs on the little membrane. And now we've turned it into a fluid movement. It's going to go up. It's going to cross across somewhere, wherever the flexibility is. And then it's going to come back out the round window and escape. The pressure escapes down the eustachian tube or the auditory tube. All right, the pressure waves distort the basilar membrane. When it distorts the basilar membrane, we basically bang the little receptor cells into the tectorial plate, which bends the hairs, opens up my sodium gated ion channels. Boom, my signal goes down my nerves. That gets relayed to the central nervous system through the cochlear nerve or auditory nerve, which becomes part of the vestibular cochlear nerve. And this shows our crossovers. So here came our fluid up through the vestibular or the vestibule, scala vestibule. It crosses across, the fluid crosses across here. This is my basilar membrane, and it goes out the bottom part, out the round window. Does that feel pretty good? You guys feel pretty comfortable with it? And this shows some different locations of where we have flexibility. So you can see some different areas, how far up the cochlea. They've uncurled it here, kind of like I did here. And this shows the basilar membrane distorting, which then is going to wiggle it to hit the tectonic or the tectorial membrane. And that's it. Isn't that ear amazing? I think it's amazing.
The thing's so small, yeah.
I know all that stuff
And
it
the
does. Yeah. All right. Do you guys have any questions on that? Do you feel pretty comfortable about it? All right, you guys, I am going to turn you loose then. I hope that you have a fabulous weekend. And we will see you guys